Hi everyone, I'm Julie Gertzley. I am founder of Rare New England. Could you tell me if you have heard of Rare New England before this conference so I can get an idea? We still have a few, okay. We are a nonprofit patient advocacy organization about a year and a half.
Just hold closer to your mouth.
About a year and a half old, and we are a group of stakeholders, doctors and nurses, bioethicists, social workers, and patients, caregivers, and such who came together to form this rare disease organization because we recognize the difficulties and the obstacles that patients with rare diseases go through every day. We have a mission with 3 components. One is to provide education and information through things like the conferences, webinars, and any other avenue that we can find. We like to create support opportunities to bring people together who are living and working in the rare disease community because we know that it can be very difficult when we're facing a rare disease and our family doesn't know about it, our neighbors don't know about it, but being in the same room with someone else who understands, we get that feeling. It's like the relief in the room. So we try to bring that forward, and also to highlight resources, because we know there are tons of resources out there, maybe not enough, but there are still a lot of resources out there. Patients, families, even physicians, and others on the medical teams really don't know about all of these resources. So we try to highlight them as we go, and that's why with a conference such as this, we're very thankful to be able to partner with UVM and Dr. Leah Burke to be able to offer this to you in Vermont. In the past, we've had conferences in Massachusetts, but now that we have Rare New England, we realize we need to get out of that state and into other states. So I'm also excited. This is my very first time in Vermont, and it's beautiful up here, and I appreciate the opportunity to be here today. I just wanted to go over just a couple of things. I want to make sure that everyone knows about the evaluations in your packets. Very important, if you could please fill these out and give these back to us at the end of the day, because your input will help us to create better programs as we go forward. I also want to make sure that everybody is aware that pictures are being taken and videos are being taken. So if anyone has a problem with pictures or videos, please come and tell me as soon as possible. And we'll respect that no pictures or videos are taken with you. Some of these will be put up on the website for later so that there'll be resources for others. I also wanted to share for a minute about how many of you are active in legislation? I know that legislation is important in the rare disease community. All right. Yes. Yes. I'll tell you, I am a nurse. I worked in a hospital. I never paid any attention to legislation in the past. Politics is not my thing. But when I got involved in the rare disease community, I do see that to be involved with legislation, to make a connection with your representatives and your senators and share what's going on in your lives, the impact from having a rare disease, that's very, very important. A few years ago, we put forth a bill in Connecticut to form a rare disease advisory council, and that was passed a couple of years ago. And that these rare disease advisory councils are to bring stakeholders together, doctors, patients, caregivers, Department of Education, anyone who's being touched or living with someone with a rare disease, and being able to come to that same table and talk about the problems that are happening in the rare disease community. Instead of people being in separate corners, it's everyone coming together and then trying to find solutions to fix the problems. So that was passed in Connecticut. It's coming along. It's been a very, very slow process, as legislation always is. We do have the bill in Massachusetts. It's still sitting in the Public Health Committee. We're trying to move that through. I'm sharing this with you because we have the possibility of bringing something like this through Vermont. So if this is something that you are interested in, I'm hoping that you'd be able to come to me at some point today. I do have some copies of the Massachusetts bill and the Connecticut bill that can be shared. I also have some papers here that really, it's already written out, just saying that you would be interested in having something like this in Vermont. If you could sign it, we do need your email address and we do need your addresses, because these are things that would be going to, as we reach out and try to find representatives and senators to back the bill in Vermont. So I can call a representative in Vermont. I'm from Massachusetts, I'm one person. They're probably not gonna listen, but if we can get signatures, get a bunch of people, 20, 50 people to sign this, then we'll have better chances of getting someone to sponsor this type of bill. All right. Any questions on that? Right. In your package, you notice you also have a couple of articles. One of them is the emergency preparedness. And I just wanted to share that that's there, that there's tips about what you can do to prepare for a trip to the emergency, things that you might not think of. Nobody wants to think about going to the emergency room. But none of us with a rare disease like to go to the emergency room. So if we're better prepared ahead of time, these are tips of things that we can do. Things like registering with your local fire departments, putting in protocols. They'll actually scan your emergency protocols into the computer. So before they even get to your house, they'll have that information. That can be very helpful. And there's also an article about psychological profiles between Munchausen by proxy perpetrators, medical child abuse victims, and the rare disease community, because some of this profile, some of it is pretty much the same. And I think if you read it, it'll make better sense. The article that I wrote originally, this one was written purposely for an organization that was covering mitochondrial disease. So that's why you'll find mitochondrial disease in there. And I am in the process of widening that to reach the whole rare disease community, because these problems do happen all over. So if you have any questions about that, I'm here all day as well. And I do want to take a moment to just recognize our sponsors for today. We do have Santified Genzyme, Shire Pharmaceuticals, GeneDx, and Invite, who have made this day possible for financially to be able to bring things together in the support, for the meals and the facility and everything else. So we're very grateful to these sponsors. And I think that's all I have. Thank you.
All right, so we're going to get started with the program. Our first speaker is Dr. Ben Solomon, who is a clinical geneticist and still sees patients, but he is also now the managing director of GeneDx, which is a genetic testing company. And he's going to talk to us about genetic and genomic testing, a little bit of what that involves. So Dr. Solomon, welcome.
Thanks, guys. Let's see if I can get this to work to start. Okay, here's lots of disclosures that I'm supposed to put up there. I think my real disclosures, or maybe the important ones for the purpose of this, are number one, I grew up in New York until I was about 11 or 12, so when I get nervous or excited, I tend to talk really, really, really fast. So if I'm talking too fast for human intelligibility, just raise your hand or throw your coffee at me or something, and so that's number one. Number two is, if we don't cover everything in these slides, I promise I will end by 10 o'clock. I think that's my allotted time, but we don't have to get to the end of it, so if you guys have questions or comments, that's great. Just please stop me. And along those lines, I don't want you guys, again, to look like this, so this can be as interactive as you guys want. I used to think this was the closest med school to where I was at the NIH, and the closest med school was across the street, the military med school. And I used to think it was that, but this is actually the Naval Academy, someone told me, who had went there. And I think the funniest thing about this is a real class. The funniest thing isn't all these folks, but it's that kind of, I don't know, zombie person right there who's just staring. And so I've always wondered what she's thinking. So the point is, I don't want you guys to look like this. And I guess the last real disclosure is I'm not going to cover 100% of everything. And obviously, you guys have wonderful geneticists and genetic counselors here, but I'm just going to touch on some topics I think might be of interest. All right, so just because it's first thing in the morning, I thought we would start with so the medical professional, the geneticists, you guys aren't allowed to say, and we'll give the first shot to those of us who don't think every day just about genetics. So I start with some Google images. Does anybody know what this genetic concept, what this is supposed to be showing? These are all real Google images. Any ideas? So this is Mendel's peas. So Gregor Mendel was this Austrian monk who figured out kind of the laws of heredity and kind of how things get passed down from one generation to the next. There's lots of interesting stories about him. Among the many stories, so one of the things he did was teach to students. He was a monk, but he also taught kind of biology. But his real passion was counting these peas, doing these experiments to figure out heredity in plants, and those applied to humans. And so he would keep all these dried peat plants in his pockets of his coat, and it was cold in Austria, and he would throw them at his students when they fell asleep. He really would. So I won't do that, but all right, let's see, next one. Oh, if I can get this to go forward. Oh, let's see. There we go. We'll do it the old-fashioned way. Does anybody know this one? So this is Rosalind Franklin, who was one of the main people who figured out the shape, the double helical nature of DNA. She did this fancy thing called X-ray crystallography that I don't pretend to understand. But there was honestly I would say a lot of us think that she was unfairly not given nearly as much credit. Oh, thank you. Okay, as much credit as she deserved for this. And then this is kind of out of focus. I'll just tell you guys. Did anybody know this one? This is kind of a tough one to figure out. So this was about the 300th or 350th. My math is probably wrong, but anniversary of Darwin's Voyage of the Beagle, where he saw the finches and figured out kind of things that helped us understand evolution, and obviously that ties with genetics a lot. And then anybody, this is a tough one. This is Nettie Stevens. She was a scientist down in Philadelphia, in the Philadelphia area at Bryn Mawr College, and she was the person that figured out kind of X and Y chromosomes and why that kind of makes us men or women and that kind of stuff. She sadly passed away pretty early in life, early from cancer, and it's likely, we know, but she hasn't been tested, but there's a good chance she had one of the kind of the common breast cancer genes, like you might have heard Angelina Jolie talking about. All right, so with that, I'm gonna try to cover more or less, I don't know, kind of 3 overlapping topics. So number 1, we'll talk about just some history of genetics and the field of genetics, and some definitions, just so we're all on the same page. Number 2, what tests do labs like ours, and certainly labs like others, what are the different testing options? There's a dizzying number of different tests that can be done. And so we'll just kind of talk so we know what those do show or can show and don't show, and kind of how those have changed. And then finally, just some kind of clinical examples and challenges. And I will warn you again before I start, there's a couple kind of medically images in here, so they're not too, but they're medically, so I apologize for that. All right, so we're gonna start first, history and definitions. So I won't go through this whole boring chart, but I just want to give kind of a timeline. Everybody, I think we all hear tons in the news about, genetics this and genomics that and precision medicine. And there's a lot of emphasis, but this is not a new field, this field of genetics. So I don't know what all of you guys do for work, but I guarantee it was better than these guys. So this was about 70 years ago. These folks figured out kind of that DNA was the molecule of heredity. And they did these awful experiments where they basically took 70-gallon drums or vats or whatever of macerated, mushed up cow heart, and they did all sorts of chemical experiments to get down to a few molecules of this substance that was DNA. So it sounds, and reading about it sounded like it was a really painful process, as you can imagine. So about 60 years ago, Watson and Crick, they published the famous, this is the shape, the double helical nature of DNA. It was a 1-page article, and one of the most important things in science that let us start understanding what was going on and find more and more answers. Certainly an amazing accomplishment. Rosalind Franklin, I showed you before, didn't get nearly as much credit for this as she should have. One of them, and I won't mention which one, but went on to say some rather awful things about genetics later in life. You can Google that, and other things as well. So at that time, the causes of genetic disorders weren't really known at all, and we didn't even know how many chromosomes we had. But starting in the late 50s, that's when we first started knowing what the causes of different genetic conditions were. So this is Pierre Lejeune. He's a French cytogeneticist, and so he figured out, kind of looking through the microscope by looking at chromosomes, the first causes of certain genetic conditions, like some of the conditions if you have an extra chromosome or a missing piece of a chromosome. And there's Pierre Lejeune with a young man with Down syndrome. What's really interesting, if you read, I can't read French in the original, but if you read the translations, he had a lot of the same bioethical kind of questions and quandaries that are still being talked about today, where we can sequence a genome really quickly. It's very the same themes continue to emerge. So fast forwarding many, many, many years, in 2001, the first human genome was sequenced, and we'll talk about that a little bit later. And it was this giant project and this giant race between, on the private side, Craig Ventnor in the front, and in the public side, the guy who's now head of the NIH, Francis Collins, in back. So I'll tell one little anecdote about myself. I was one of the first kind of big talks I gave was when I was in training at the NIH, and it was like this project that I'd done for 6 years and just painstaking work, and I put my heart and soul into it. And it wasn't that exciting, but I was really proud of it. And so I had the chance to talk in front of lots of people at the NIH, including Francis Collins, who was sitting, if I'm standing right here, he was probably sitting where you are, like that close to me, like right up front. And I was so nervous and my heart was pounding, but he spent the entire time on his BlackBerry just like texting emailing people. So I don't think he heard a word, but anyway, I got to speak to him. So, and then again, recently, I don't know if you guys saw in the news, but the first time in the United States that someone did this new technology called CRISPR, where they're able to change in humans, a genetic change in someone that led to a genetic disease. This is a guy at University of Oregon who did that. Again, this is very early in this. It doesn't mean that all of a sudden that there's going to be these brand new therapies like that'll work, and there's tons of questions. But it's the first kind of things saying that, okay, yeah, it maybe will work. We should keep thinking about, can this work, should this work, et cetera, et cetera. All right, so one of these really boring textbook pictures, but again, I'm just going to kind of just to define all these things. In each of our cells, of course, we have all our DNA, our genome, and it's packaged onto these different chromosomes, right? These things that are, anyway, that kind of funny little X-shaped thing. And so these are made up of all the DNAs on there, right? And so we're talking about a lot of DNA because that's what I think about a lot. How do we look at the DNA and try to find answers? But then the DNA is made into RNA, and then the RNA is made into proteins, and the proteins is the thing that are kind of doing the job, right? So, but we talk a lot about trying to find the answers by looking at the DNA. All right, some more definitions. So if we talk about all our genetic material, right, that's our genome. So each of us gets about 3 billion letters in each cell of genetic code from mom, and 3 billion from dad, roughly. Actually, moms give us a little more because they give us some extra things. But about 6 billion total letters, right? And of that, about 1% of it is the protein coding portions of the genes, right, called the exome. Okay, about 1% of it. But it's estimated, nobody really knows, but we kind of think about it and we guess, and based on kind of the, some good math, maybe about 3/4 or so of disease-causing mutations, especially for rare conditions, these Mendelian conditions, about the majority are found in that 1%. So a lot of us really focus on that. And then we'll talk later about why we also focus just for practical reasons on that. Okay? All right. So there's a crazy thing is that we don't even know, so I spent, and Dr. Burke and Dr. Wilden, we've spent, and all of us have spent our careers in genetics. We don't even know how many genes there are. I can't give you an honest saying this is exactly how many genes. We estimate maybe around 19,000 or so. Of those, about 4,000 or 5,000, depending on how many you believe, are known to, if you have a change in one of those genes, you can have a Mendelian condition, a rare genetic condition. Because of these new technologies, there's lots of new discoveries being found. So right now, there's 14 new disease genes being published per month, mostly because of these new techniques. So 14 new disease genes published per month. And it doesn't even count all the ones that have been identified, but are still, in the publication process and so on. So things are really, really going at a dizzying rate right now. Again, these are, I just want to emphasize, so these are, when I talk about these Mendelian conditions, these are conditions where one genetic change in one gene, right, can give the condition, or maybe it's one genetic change from mom and one genetic change from dad, the combinations of these are Mendelian conditions, as opposed to these multifactorial conditions, and these tend to be common conditions, right? Let's talk like ADHD or diabetes, where we think there's some genetics going on, but it's really complicated. There's tons of these little genetic changes from mom, tons from dad, tons of non-genetic factors. And so I'm not talking about those. So it's estimated, and these are all just kind of estimates and hypotheses, that about 80% of rare diseases are these types of Mendelian conditions. We don't know the answer for all of them, but a lot of them are, you know. And I'm sure you guys might disagree with some of my estimates there. These are just the things that I've read and kind of I think are right. But, you know, the textbook is constantly being rewritten. All right. And these conditions affect everybody from all walks of life, from humble folks to the most, you know, famous celebrities and royalty. So Queen Victoria, you know, in her family there was hemophilia, a rare disease. Do you guys know Stranger Things, that really creepy Netflix show? So I watched that. I didn't sleep for weeks, but I watched it. But one of the stars of that show has a genetic disease that affects his teeth and some of his bones. One of the best basketball players in the world right now has a condition, a ciliary condition, so that his organs are arranged differently. So they affect lots of different people and from all walks of life. They've also been around since for all of human history and before kind of recorded human history. And this is one of the, I apologize, kind of the graphic pictures. But this is a skull of a person who had a skeletal disorder that was found in Spain about 500,000 years ago. And from examining kind of the skull and the bones, it was clear that this person had a severe, maybe genetic, but a severe skeletal rare condition and would have needed a lot of extra help, right, because of that. And the fact that this person lived long enough, right, that this person lived so long is evidence that this society 500,000 years ago was taking care, you know, of folks that needed these, that needed help with these kind of things. And it's a nice thing to see, you know, 500,000 years ago. Jumping forward still in ancient history, but more recently, you know, it's thought that King Tutankhamun, King Tut, had some type of genetic disorder, again, probably affecting his skeleton. They know that from the shape of, you know, his sarcophagus and the shape of his body. It's also very sad. He passed away at a young age, and you know, clearly also had a lot of infectious diseases like malaria and, you know, malnutrition, and so he had a tough life. Okay, so going back to this, you guys are already sick of this picture, but just the point is that we're going to talk now about more about DNA, and a little bit we'll touch on the other things later. All right. So in ancient history, you know, if I wanted to look at someone's DNA, or someone went in to see the genetic counselors here, the geneticists here, 15 years ago, 20 years ago, the only way to look at a gene, a single gene, right, would be this kind of thing. So this is called Sanger sequencing, or bidirectional dideoxynucleotide sequencing is the fancy name, okay? I always get, that's good. Usually I get my tongue tangled up, but today I said it without tripping. So if you wanted to sequence a gene, it was a slow and laborious process, right? Because you would take, say, I want to look at this gene. So you look at this piece of it, you'd get the gene, you know, the person's DNA, you look at this chunk, and then this chunk, and then this chunk, and then this chunk. And so it was all kind of in series, you know, it was a slow and painful process, and some genes are really big. So the game changer, or the thing that's changed so much in genetic testing now, is this. And I won't go through it in gory detail, but the basic idea is that we can take someone's DNA, right, split it up into lots of little pieces, grab all those pieces, and sequence them simultaneously in parallel, right? So we could say, okay, let's look at 20 genes that might cause a certain rare condition, or let's look at the whole exome, all the genes, right, because we're just not sure what's going on. Or we could look at the whole genome, right? And there's pluses and minuses. It's not perfect, but it certainly changed a lot of, you know, when I see patients, how I practice the ability to do this, okay? So I want to stop for a second. I've talked way too fast, I know, and I just want to ask if there are questions or anything, or is everybody just so dazed into boredom? Yes, sir. Yeah.
So you come from a company, you attended a company that does this gene testing.
Yes.
Are you in Vermont?
No, so I actually lived in Vermont. We are based, because a ton of us came from the NIH, so we're based in Gaithersburg, Maryland, but we test folks from all over the country, all over the world. Melissa, who's up there, is our area rep for the company, and so we work all over, yes.
Okay, thank you.
Yes. And so anyway, these new methods have led to the ability to make genetic testing much more available, much cheaper, right? And so you can see basically the cost per genome going way, way, way down. It's because of these new methods. These next generation sequencing are massively parallel methods. So that's just the cost for sequencing, though. You still have to think about it and analyze the data and figure out what it means, right? But just the cost of generating the data is a lot less. So again, to put things in context, so in 2001, when the Human Genome Project was finished, right, that first human genome cost about, depending on what you thought of as a cost, between $2 and $4 billion, right? It took over a decade to do. It was done in these giant rooms. Imagine like rooms all over the world in 20 different centers, like the size of this, with these machines going day and night to get, honestly, kind of a lousy version of the human genome, right? But they were using different technology. They did a great job. It was amazing. They invented all new things, but it's just, they didn't have the same tools that we have now. So now, using a machine, actually, we have one in our lab that looks just like that, called a NovaSeq, just this new machine. For about $1,000 in a couple days, using a machine like that, right, we can get a much better version. It's not perfect. Everything's not covered, but a much better version of the genome. You still have to analyze it. You still have to interpret the data, but you can get much better data and much faster and much cheaper data. I don't have a crystal ball. I don't think we're gonna you're gonna spit into your phone and it's gonna say bloop, bloop, bloop. It's not gonna be like you have this. I don't think it's gonna be that. But my guess is that as the technologies get better and change, it'll be much cheaper and faster, and hopefully also, I think really importantly, we'll be able to get a better look at the parts of the genome that are hard to see, because there's certain parts that are just really difficult to look at and analyze and understand. And so lots of good things coming down the pike, and the future's exciting in this area. And so a lot of that excitement led to things like, it's now called the All of Us Initiative, but the thing that President Obama started, where the idea was to sequence lots of people so we could basically just understand genetics and how it contributes to both rare disease and more common conditions. Also led to things like the Cancer Moonshot, right? Again, a lot of it's based on if we can understand genetically what's going on with cancer, rare cancer, common cancer, we can figure out how to manage it better, how to treat it better, and how to help people that are affected by these conditions. All right, so again, we're going through kind of boring, this hopefully doesn't feel too much like a bad flashback to high school biology, but just more boring definitions. So just to give you a sense of the scale of things, so one genome, if you were to print out each letter of one person's genome in a book, it would fill up 2,000 War and Peaces, right? And that's a book I've never gotten through, War and Peace, but it's a really giant book, right? So 2,000 of those, that would be the size of your genetic code. Another way to maybe look at it is this is someone's genome that we sequenced. And to give you the sense of the amount of changes that we see. So if we were to sequence any one of you guys in the room, right? Not necessarily looking for anything, but just saying, okay, how many different genetic changes you have, we'd find millions of different genetic changes, millions and millions. Lots of them, we would say, oh, that doesn't mean anything. A few of them, we might say, okay, that is the cause of the rare condition this person has. And a lot of them we would say, geez, we just don't know how to interpret those yet, right? It's really is, it's really exciting. There's great things, but this is a relatively new science. So I guess the point is that, people talk about looking for a needle in a haystack, but maybe a better analogy, when we're looking through a genome, we're looking for the needle in a lot of other needles, right? So it takes a lot of very smart people, and I've been at GeneDx for a year, and my first thing to do, one of the big things, is I invested in lots of really smart people. They already had some, but really good, basically bioinformaticists, which are computer programmers who know how to do this, and we do all sorts of very cool things with machine learning to pick out these things and help the humans figure out the answers better. But again, I don't want to be too much of a wet blanket about all this. There's still a long way to go. So this is a project that is still ongoing. And so remember I said about 1% of the genome is the genes, right? The protein coding portions of the gene that's doing the real job. And the rest of it, we don't really know what it does, most of it. It's thought, oh, maybe it's left over from when we evolved from, I don't know, trilobites or woolly mammoths or whatever. That's not good for evolution, but you know what I mean. And so these guys did this project, and they looked at all that other stuff, and they found that 75 to 80% of it was biologically active. It was doing something. We don't really know what it was doing. We don't know kind of what a change in it would mean, but it's doing something. And so we have a long way to go. And as my previous boss would say this, and so I stole this idea from him, is that genetics and genomics is so cool, it's so great, but we really are still at the Wright brothers phase. It's not like it's a solved, we can understand everything. We're early in the stages of that. And then again, I want to be very honest about that. I think it's important to realize that there's things that I think are amazing that we can do with genetics and genomics, and diagnosing folks with rare conditions and finding better answers for patients and for families, and there's so much cool stuff. There's also, on the flip side, a lot of used car salesman mentality. I'm not going to pick on specific, products or companies, but there's a lot of hype around genetics and genomics, and some very good business people are really good at taking advantage of this. So be careful or talk to your local expert about some of these things. So yeah. There's lots of great, by the way, if you do Google images on used car salesmen, there's so many funny things. All right, so I think most of you guys know what a geneticist does, but kind of in my idea, the reason that I love seeing patients and why I think it's really exciting to me is it's these questions, and it's really exciting to try to find answers for folks. So one takes information right from physical exam, from family history, this is a family tree or a pedigree, and then sometimes from imaging or labs, that's someone's part of their aorta, right, one of the big blood vessels, right, and tries to figure out the answers, right, and therefore what to do about it. I don't think this, does anybody know this show? Is this, so House, yeah, so it's a lot like, a real life version of this, hopefully without all the drug use and affairs and all that kind of stuff, but it is, it's a lot, sorry. And therefore, what can I understand about this person with this condition, this family with condition, and what can I do about it? And so along those lines, so one has made a diagnosis. We don't always figure out diagnoses, but sometimes we do. So what? What do we do with that information? My wife is an OBGYN, and she's running around seeing, doing like 100 C-sections a day and seeing 1,000 patients, and just, it's amazing how much she does. And I'll see, 3 or 4 consults, in the afternoon, and I think I'm so proud of myself, and I'm so busy, and she doesn't give me any respect for this. It's a different thing, right? But anyway, why does it matter to make a diagnosis? I don't think, I'm preaching to the choir, hopefully, but I think there's a number of things. There's, information for families so they can understand and have better knowledge and sometimes stop, wondering, right? Just, why? And sometimes this is a guy who played for Baylor, a basketball player who had a condition, a connective tissue condition called Marfan syndrome. So we know that, he should get ultrasounds of his heart every year, right, to make sure he's healthy. He should be on certain medications to protect him. And this, part of my language, this sucks, but he shouldn't play basketball, right, because he puts himself at very high risk, right, of something bad happening. And then sometimes by understanding molecularly, biologically what's going on, we can develop new treatments. So I can't take any credit for this, but one of my favorite mentors is a guy at the NIH, Bill Gall, who works on lots of biochemical conditions. And so these are pictures of someone's eyes, and so he works on a condition, among other things, called cystinosis. And so in cystinosis, sorry, that's tough to figure out what's going on, but see that little thing that has an X over it, right? That's kind of a channel that's going out of part of the cell. And so cysteine can't get out of part of the cell, and so you have these crystals build up in the eye, and so folks with this can go blind, right? It can build up in the kidneys, so it can be bad for your kidneys. It can build up in your thyroid gland. So by understanding kind of what the gene involved is and what the biology under it was, they were able to figure out basically a way to treat it, right? So eye drops that help get that chemical out of that part of the cell, and oral medication that one can take. And this doesn't happen for every condition right yet, but it's understanding what's going on helps lead to development of new treatments and therapies. All right, so what tests can a lab do? This is now the second part of the talk. And so you guys are gonna get really sick of this analogy, but this is the analogy I always use of kind of like looking at someone's genome or in different ways, right? Is like looking at the world. So I use this example of a world map. So one of the older but still a really good tests. Does anybody know what besides the genetic professionals, what this is called?
Oh, I forget. I did this in college. I used my genetics test. I could tell you and everything, but
So no, you just said it. So these are chromosomes. That's perfect.
The chromosomes, and on this certain line, who has the extra chromosome, whether they're the ones that affect gender or ones that affect Down syndrome.
Yep.
I remember being given a printout of 1,000 of these all in black, and I'm going to cut each one out and find out which one was the chromosomal abnormality.
So you're like an advertisement, not just for genetics, but for computers, right? Because now computers.
So when you were going to ask if there were any questions, I did have a question about that.
Okay.
Because I started out as a biology major, but then my math and chemistry skills didn't back me up for pre-med. But anatomy and physiology and genetics, if it was based on those two things alone, I would have been okay. So my question was, today, 20 years later, than when I was in genetics, are there computer systems where you can send through your lab results that can pick out like an abnormality or an extra chromosome?
So it's such a good question. So yes and no. So there are ways that I believe very strongly in this, that the computers can highlight things for us to look at, can maybe, so you don't have to cut out all the chromosomes, can arrange things, right? But especially with all, exomes and genomes, computers are good at arranging data, but the databases, the information sources they bounce off of are great but imperfect. So we strongly believe we've invested in, you got to have computers that are helping you, but at the end of the day, you need deep human clinical and molecular knowledge to say, okay, the computer thinks this, but I need to double check it. I need to figure out if there's something else going on that the computer doesn't check, right? So, and I think we're getting more and more there, but it's certainly not like we're not Terminator land, nor will we ever be, right? Yeah, yeah. But they're really helpful tools, I would say, but you can't over rely on them. Yeah. Yeah, that's okay. So this is a karyotype. These are chromosomes, right? And just again to give a sense, remember we talked about 20,000 genes. A good karyotype will have about 60 bands, 60 stripes on them, right? Those they look like kind of zebra stripes. So that means if you do the math, there's maybe like 30 to 40 genes per stripe. Okay, so just kind of keep that in mind. This is a fuzzy image. What karyotypes are really good at, right, is not only seeing, okay, how much of things are there, like the sex chromosomes and the chromosome you said for Down syndrome, those things, but kind of where it is. So these red arrows are showing you that two pieces of these chromosomes just kind of switched places, right? So you can see that, or the computer can help you see that, okay? But remember, you're like 30 to 40 bands per chromosome. So when if you see something on karyotype, it's a really good test, but it's usually a pretty major thing, right? Like something affecting, the whole half of North America, right? Because just the scale of it, you're looking at things from maybe like a 50,000-foot view. You can't really zoom in enough, right? But still a really good and useful test in lots of contexts, prenatally, for cancer, for lots of different reasons. Sometimes, of course, if you guys are coming in to see the good geneticists and genetic counselors here, sometimes there'll be a tip-off from a specific thing that says, okay, I'm not going to do a broad test, right? Not just a karyotype or something that looks at everything, but from a certain hint, and this is just supposed to remind me this is someone's heart, a certain type of heart difference that a child might have. You can say, okay, let's do a targeted test, right? So the first ones of these targeted tests came along, and this was an old but still a really good test. It's called FISH, right? And it's nothing to do with like the swimming fish. It just is an acronym. It stands for fluorescence in situ hybridization. So if someone with that particular heart condition came in, they might say, okay, I'm not going to do a whole karyotype or some other test. I'm just going to send in this probe. Sorry, I'm not tall enough. That little red thing. I'm going to see. I'm guessing there's something different about that one piece of the chromosome. So I'm going to do that. Look for that one probe and see if it's different than usual. Okay. And again, this is more of an evolution or a kind of the way things have changed. So smart people figure out, okay, what if we take those important probes and just kind of stuck them all together on one test and looked for all those things simultaneously? And so, sorry, we're going back. So looking at a probe is kind of, I told you, I'm going to do this analogy to death. So then you're able to do something smaller, you're able to zoom in, but still a pretty big thing. Okay, does that make sense? But anyway, they took all the important probes and they stuck them on one chip, one tile, and it ended up, right? And they got more and more, they got better and better. And now these probes, like we just talked to a company that makes, these probes, you do like 7 million on a single chip, right? So can someone tell me what condition this person has? No, I'm just kidding. I can't tell. So the computer might be able to tell you that, but you can see. So this is called a microarray, right? And it's a really good test to see, okay, are there areas of DNA where there's extra or areas that are missing, right? That might be clinically important to have extra or missing DNA, okay? Again, not a perfect test. It doesn't spatially tell you where things are like the karyotype does with the translocations, but a really useful kind of first-line test for lots of possible genetic conditions. When I see patients in the NICU, it's often one of the first tests that I do, when I'm not sure what's going on, but I think this is a good test to start with. Again, we're zooming. So I'm picking on where I live. I live in DC, so we're zooming in more and more, but you're still able to see things, but you're not kind of on the gene level yet, usually, though sometimes you can pick up part of a gene, okay? So it's a microarray. Again, we're just talking about ways to kind of zoom in and look at more and more of someone's genetic information. If we want to go down to the house level, and this is kind of my neighborhood, right? I live right in the middle of DC, right? If you want to go to the house level, the analogy we're using is the gene, and there's different ways to do that, to look at the individual of the 20,000 genes. One way is Sanger sequencing, okay? And again, it's a good thing, and it's a really good, reliable technology, but you can't do a lot of it at once, right? It's mostly for like, okay, let's look at this chunk of a gene or this whole gene, right? Or you can do this next generation sequencing, right? and this is where you're sequencing lots of stuff at once. This was the very first exome I did when I was at the NIH, a research exome. So again, you're chopping up all these bits of DNA. The computer is lining them up, okay? And then you're seeing, okay, these different little, these changes. So wherever there's this, that's the same base pair, you're just getting lots of different looks at it. And so this means that on one copy of that gene, a person has a change, and on the other copy, you don't have a change. Does that make sense? That's all this is telling you. So the genetic term for that would be a heterozygous change. You have one copy with the change, one doesn't. It doesn't really matter if I don't know why I belabored that. But anyway, so this is looking at the same chunk of DNA. This is through the good old-fashioned Sanger sequencing. And above it is next generation sequencing. So you have a change right here. Okay, you have one copy with a change and one without the change. And that's just different ways that you look at it. Okay? And so I talked a lot about DNA so far, but also want to make the point that DNA is not the only way to try to find answers. Okay? We can also look at biochemical testing through newborn screening or other ways where we do biochemical tests to look for certain conditions. And then I'm very biased. I really like the sequencing, obviously, and the testing, and kind of that's what I think about every day. But there are certainly times that I'm sure that a geneticist or genetic counselor would tell you, like, look, I don't think genetic testing makes sense now, right? I don't think it's necessarily going to help us or might not help us find an answer that's useful. Or maybe we can, based on things on physical examination or x-rays, or this is a biopsy, right? We can make an answer without needing to do genetic testing. There's certainly instances. So it's not the end-all, be-all, just genetic testing every single time, right? And that's why you need very smart clinicians to guide you through this kind of just dizzying number of different testing options. All right, so I'm still amazingly on track. I'm going to go through my third area, which is just some examples and honest kind of more challenges, what's difficult. Again, I want to pause and see if there are any questions or comments or anything from anybody. Yes?
Obviously, there are a lot of places that are doing testing. Does the information get shared? So that
Yeah.
Can you repeat the question?
Oh, yeah. Sorry, that's such a good question. So the question is, are there lots of people are doing this? Is the information shared? I think it's a really important question. And so there are several ways that's shared. I'll talk about what we do, okay? Because we share, I think, more than anybody. So there are public resources that are funded by the NIH, like ClinVar and ClinGen, where anything that we find that's meaningful, right, we deposit it. So anyone in the world can click on that and say, this genetic change was found by GeneDx, and this is our interpretation of it. Was it, did it cause a condition? Did it not cause a condition, right? And we have a pretty sizable team that just focus on doing that, right, with molecular geneticists and genetic counselors, right? Some labs are better about that than others, and it just takes resources, and you just have to believe that this is a good thing to do, which we do. We're very proud we are the #1 clinical lab contributor to this. The other way that we do this, a lot of what we do, for example, through exome and genome, we are constantly finding, okay, here's a new genetic change in a kid. It kind of fits, but I've never seen anyone else in the world with that. I don't know, could this be a coincidence or not? So we do something, for example, we're the biggest contributor to something called Gene Matcher. So we put it out there, we just say this gene, if anybody in the world, another lab, a researcher, we collaborate with tons of researchers and clinicians all over the world, has ever seen the same thing, it just automatically matches us up, right? So we can say, okay, here's a child, and this just happened, here's a child from Australia who has something that's different about they have a skin condition. And here's someone in New Jersey that has that same condition, right? So we can match those clinicians or the researchers up together and do that. I would say, despite that, though, it's imperfect for a couple of reasons, right? Number one, the data sets don't always match up perfectly. Maybe like one testing platform isn't the same as another, so you have to be careful about how you interpret it. And number two, there's way more stuff out there than is shared. And I'm not going to name names, but some companies refuse to share, right? They just think, from a business proposition, it doesn't make sense to share, and that's tough. And there are researchers who are wonderful, but frankly, they don't have the resources to do this, right? They're killing themselves to figure out, what the cause of this is, and it takes a lot of time and computer savvy to do all these things. So yeah, so one of the things, I guess one of the points though is that as rare disease advocates, one of the really important things I think is to advocate for sharing data, both clinical data, both genetic data, and I would say that's made it's night and day between now and even 5 or 10 years ago, how much data is publicly available. Does that answer? Yeah, thanks. Yes. No, no.
Oh, sorry, thank you. You were speaking earlier about how genetics, you're inheriting them from both parents. So a lot of times you want to talk to families when looking into, do you run into problems with HIPAA? Or, I mean, is that hard when you're trying to go back through families? I'm with Angel Flight Northeast, so I'm here as a resource. So that was just a curiosity when you're.
Yeah, Dr. Burke, do you want to address that maybe? Or I would say yes, there are always situations that are very challenging. There are ways that we have to obey compliance and legal and HIPAA rules, and we often run up against challenges, and we try to manage them as best we can, but there are difficult situations.
Yeah, I think one of the points I would make is that when he says that they're putting in clinical differences as well, that's de-identified clinical differences. So it means that they have the genetic change, and they're saying this genetic change was seen in a person with these features.
Yeah, I'm so sorry. So and it's the genetic change in isolation. It's not like, hey, here's a genome of John Smith, and here's everything about him. It's just this specific genetic change went with, let's say, this heart condition, right? Okay, yeah, so thank you. Yeah, I can see how that one would go over very terrifyingly. Yes, that's a good point. Yeah, yes.
So I was at a conference back in March, and I was really struck by this company called Genomic.
Yes.
They had great swag,
Yep.
which was nice.
Yep.
But then it got me thinking, like, how accurate is that kind of facial recognition for diagnosing genetic diseases?
Okay. So I'm going to, I think it's probably on my slide. So I'm on their scientific advisory board. So as a full, but I use them as a clinician beforehand. So I think it's thank you. I think I need a new prescription. So the facial recognition is very cool. It's absolutely imperfect. It works super well for certain things, and other things it doesn't work well at all for, honestly. And I remember, for example, with their platform, which is great. There's one condition that constantly comes up, and I no matter, I say, I know it's not that condition. There's no way. And so it's one of these things that data sharing through that kind of thing will make it better, right? Will it ever become a truly useful thing? I don't know. I don't know how you guys feel about it. Some clinical geneticists love it. Some think it's not a helpful thing. I think it's a neat kind of proof of concept exercise, but I don't know if it's actually that. I'm on their board, so I'm very biased, but I don't know how useful it is yet in reality, I would say. The thing I like about it is that it's a good way to collect information about a patient. They have a very user-friendly interface, so that later when we're matching up the genetic analysis with stuff, that helps. The way they do it, by the way, a question comes up with this for privacy reasons. So what they do is you take a photo of someone, and then it says by facial recognition, could this person have this rare condition or that rare condition? And so there's obviously lots of questions about privacy. They store it in a way that's not the storage of the photo, right? It's just kind of zeros and ones. It gets broken down too. Yeah. So I think it's really an interesting kind of proof of concept exercise. I wouldn't necessarily say, oh yeah, it's that face of gene said that this is the answer. Yeah. All right. All right. I want to go into some examples and challenges now. I know you can't see this. This is okay, but I think one of the ways, in a good way, that genetic testing is moving. This was a patient that I think about a lot that I was involved in. Where this child was born. Obviously, it's something very severe. Was in the NICU for a long time. Each of those boxes represents a different test that was done. Trying to figure out what the answer was. Some of it was genetic tests. Some of it was biochemical tests. Some of it was X-ray type tests. Some of it was, let's have this ophthalmologist come and try to figure that out. Until someone threw up their hands and ordered an exome, and we got the answer back immediately. And I think it's very humbling as a physician that the bioinformaticists are often really, really good at finding answers. I intentionally put this up because this was not anything that provided this family with a silver bullet that therefore, there's, here's a treatment that's going to fix this. But if you can imagine, if having the answer earlier, right, you would have avoided a lot of hospitalizations and procedures and kind of questions and difficulties, right? I think you guys have probably all seen this slide or some version of the slide before. There's some really good stuff from these guys, some really nice kind of graphics. But the point is that for rare diseases, the average time for a diagnosis is 8 years, right? Or it's maybe a little better now, but it's not good, okay? And the nice thing is that we do have tests now that can really short-circuit this process in a way that weren't available before. these tests will not provide answers for everybody. I think that's an important thing to know. So for example, when we do exomes, I'll just take our data. We find answers in about 34% of people, and we're very careful not to say, oh, we think it's this, therefore this is an answer. Does that make sense? 'Cause it's easy to take some of these unclear results and say, oh, this is an answer. But very honestly, about 34% of our times we do things like exomes, we get an answer, which is amazing that for every rare condition, a third of the time you're going to get an answer. Of those, about 9% are brand new genes that are out there where we found multiple people, but they hadn't been previously published. We do genome sequencing as well. I'll just touch on that. There's a question, if the exome gives us, 33% of answers, is genome going to give us the rest? I honestly don't really think so. It maybe give us a little bit more, maybe not a ton more, but in certain situations it might be helpful. And sometimes this can be very actionable. So we have something, and again, I'm not just trying to make an advertisement for us. There are other companies that do this, but where we do an exome, usually for critically ill patients, and we get the answer back within a week. Usually within about 5 days, we call with the verbal answer. We do exomes of families, mom, dad, and the child. And then we have a paper report in a couple weeks. And in those, we get answers in about half, okay? 40 definitive and 40%, and then new genes in about 8%. And the latest one, and again, not most of the time, it doesn't give us a silver bullet, very honestly. It gives us more answers and information, but it doesn't give us an automatic treatment. This was a kid who had neonatal, as a baby, had fractures, right? She was still in the NICU. She had fractures right away. She had something else, she was just very sick, and we did one of these. And she had mutations in this gene, IL-1RN, and there is a treatment for this. It's an autoinflammatory condition. Basically, your immune system is way revved up. So you give this treatment called anakinra, and the kid will get better, right? Which is a wonderful thing to be able to have that answer for those folks. We do get answers a lot of the times with this, right? But there's also 50%. It's a glass half full, glass half empty. There are many people with rare conditions that even with these great tools, that we still don't have answers for, right? And maybe it's because we're just not good enough at looking yet, or as we'll talk about, maybe it's not genetic. So one more point about that. So this is Max Munka. I trained in his lab and at the NIH, and I did a lot of work with him. And he discovered this condition named after him called Munka syndrome, which is a skeletal condition. And so I'm not the world's expert on this, but I saw a lot of patients with this. I wrote textbook chapters on this. I did a lot of work on this. And I was at a kind of a fundraiser maybe 3 or 4 years ago. And there's some families there and some people, some very wealthy people we were trying to get money for the children's hospital for there. And I spent a long time talking to this mom and dad, who was really nice people, and they had participated in a research study, not necessarily because of a rare condition, but because in their family, everybody had been born very early, very prematurely, right? And so they had just participated in this study. I talked to the dad for a long time. They showed me pictures of their son, who was doing really well, even though he had been born prematurely. And I didn't really think that much of it, but when we analyzed their genome, of course, what did we find? This was the only person in these many thousands of genomes we had sequenced at this research institute that I used to be part of, with the defining genetic change for Munka syndrome, right? And I spent an hour and a half talking to that dad. I saw pictures of that kid, and I did not pick up that the family had Munka syndrome, right? And I still looking back, I still don't think I would, So all these new tests, not only are they giving us answers, but it's a very humbling thing to be a physician or a genetic counselor or in this field, I feel like, because a lot of times the answers aren't something that we would have suspected. We're rewriting the textbooks about what these conditions mean, and it's a very interesting, and again, it's a very humbling time to be part of this type of field. All right, so in the last couple minutes, I talked about kind of some success stories, but I want to talk about what are we honestly not perhaps as good at. So there's this kind of, you guys have probably seen this before, this idea of a guy is looking for his keys, and someone comes along and said, oh, you're looking for your keys right here under this light. Did you drop them right there? He said, oh, no, I dropped them back in the, 1 minute, I'll make it. I dropped them back in the parking lot, but the light's much better here, right? So we tend to do that in genetics. We tend to look at what we're good at looking at, right? So we're really good at looking at the exome, We're not as good, as I talked about before, at understanding the rest of the genome, There's a lot of work to do to understand that as well. We're also good at looking at the stuff that we have. So if someone gets a blood sample and sends a test to us for a blood sample, we get DNA that represents the blood sample. Or we can do all our tests with these cheek swabs, right? So that's DNA from here, right? But maybe a person has a genetic condition where the genetic change is, let's say, just in their heart, right? Or in the kidneys or the brain, but we're looking at the wrong tissues, right? That doesn't mean that Dr. Burke or Wilden is going to order, a skeletal biopsy or something of everybody. But sometimes we're honestly, we're not looking in the right place. There's a really beautiful study. I had no part of it published a few years ago, where all these kids with characteristics, certain types of differences with their brains. Okay, they didn't find the answer by looking in the blood, but they found it right by looking bioinformatically at the blood, the DNA in a different way. And then they did some biopsies of the central nervous system and found the genetic changes just in those parts that were affected, right? So sometimes we're just looking in the wrong place again. The other thing, sorry, these are more kind of very medical pictures, but we're also honestly not really that good at looking or knowing the answers, being able to pinpoint it for these multifactorial conditions. So certain things like certain types of spina bifida, gastroschisis, which is something that can affect the abdomen, certain types of cleft lip and palate, right? These are things that are we still think, kind of like diabetes maybe, these multifactorial conditions where it's very hard to pinpoint exactly what it is. Again, this is hopefully to not make you feel too dubious about this whole science, but all these things that we're terrible at. So we're good at looking at DNA. We're not yet that good at looking at RNA or proteins to find answers, right? So we're really good at looking at exomes and genomes, but looking at RNA, that's an RNA type thing. We're not that good at looking at epigenetics. We haven't even talked about that, but we can look at some things, but things that can turn on and turn off genes without affecting the DNA. And then there's all this stuff you might read about microbiome, about maybe the bacteria and viruses and stuff that live on us. So we're just starting to understand how all those things intersect. And then this is the very last slide, I promise, because I know I'm over. But there is, again, a lot of work being done on this. And I think the really nice analogy that I've heard on these really hard things is that it's like Google Maps or whatever. And I use this constantly, or what's it called, Waze, like those kind of things that tell you where to go. And so just like they layer on, here's the topography, here are the roads, here's the traffic, here are the buildings, right? In the same way as we layer on, okay, here's the genome, here's the transcriptome, the RNA, here's the microbiome, the bacteria that live on us, here's things that affect our, our social lives and our families. As we put all those things together, and it's not going to be easy, we'll get hopefully more answers and therefore more kind of management options for everybody. With that, thank you so much. I'm sorry I went over a little bit, but I appreciate your time.